To develop a new drug, it takes a decade of work, maybe more, and at the end, it often doesn't work. Most drugs that get to a clinical trial, they don't make it to the patient at the end of that process. And the question is why? They become unsafe because they cause cardiotoxicity. So how can we identify which drugs are going to be cardiotoxic long before the drug gets to that point of failing?
Ultimately, what we would like to do is have preclinical development as fast and accurate as possible to help only test the best drugs in patients or the clinical development side. So I think in clinical development, your success is really hinged on the quality of your preclinical development, such that you're getting rid of those early drugs that are going to be potentially toxic and really focusing on those that have the best chance of success. And CARDIOVERSE is really right now improving the preclinical development side.
The purpose of CARDIOVERSE is to build a simulation platform where we can assess in a computer program whether or not a drug will be toxic. So that kind of heart failure toxicity is common for a lot of drugs, and it's not well understood and it's not particularly well captured preclinically. It's a leading reason for failure of drugs in clinical trials.
So for the CARDIOVERSE to work, we need a lot of data. That is like at the heart of what we need here. And so we'll get these complementary data sets that allow us to explore things in slightly different ways. We're going to be able to take stem cell lines from many different individuals to capture genetic diversity, and we'll throw enough different medicines that we know already cause defects and some that we know do not, so that we can establish a model of how toxic is this drug likely to be. But we can't do that with just the heart cells. There's things we can't do in the dish that only happen in vivo in the mouse, but combining the two of them, that makes these models even better than they are today.
Our strategy is to use complementary model systems. And so by measuring in genetically diverse mice, we can capture the spectrum of physiological responses that a mouse's heart will have to a drug and put it into an AI model to make a prediction of what a beating heart will actually look like in response to that drug. So it's not just, there's a 20% chance this drug will be unsafe in this individual. We want to be able to say, this is what this person's beating heart will look like in response to this drug. This is what we would predict of their clinical measurements.
We have to find the technology that gets us the best answer so that when we go to patients, the drug works and is safe. I think that if that's your guiding mission, everything else follows suit.
We have a chance here to do something at a very different scale. This is a scale where it's harder, perhaps, to get to a clinical trial for any given disease, but if we can reduce the chance that those drugs are going to fail as they go through those trials, this could have an enormous benefit that is far wider than any of the things we've done before.